It's been a really quick week, but I'm really excited about our final talk. I think it's super exciting. So let me introduce our speaker without further ado. Our speaker today is Simran Arora. She's a PhD student at Stanford University, advised by Christopher Ray. Her research really blends machine learning and systems, with recent work focusing on developing insights into what really makes foundation models work well, and then leveraging these insights to design new architectures and algorithms to make machine learning more efficient. Simran has been recognized for outstanding papers and oral awards, as well as spotlight awards at top-tier machine learning conferences like ICLR, ICML, and NeurIPS. And really, in addition to her research, Simran recently co-created the Stanford Systems for Machine Learning course, teaching more than 100 students. So I think this will be super exciting for us here. And she's grateful for support from the SGF Sequoia Fellowship. And today's talk will be on foundation models. I think it's a really nice way to wrap up this week's summer school. And thanks so much for joining us, Simran, and over to you.
Awesome. Thank you so much for having me. Let me just share screen and we can go from there.
Yes, while Simran is sharing screen, if anyone does have questions during the talk, just pop them in the chat and we'll catch up afterwards.
All right. Can you see the slides?
Sure can.
Awesome. Great. All right, let's get started. So hi, everyone. As mentioned, I'm Simran, and I'm really excited to be here to talk about my research on algorithms for efficient machine learning. And this work is at the intersection of ML and computer systems. We've been seeing many promising results in this area lately, and I'd love to share some of this with you and how these breakthroughs can help on the medical side as well. So nowadays we have amazing ML models that can do things we didn't even think was possible a few years ago, whether debugging code or generating fun and impressive photos, or starting to predict protein and RNA structures towards maybe designing new drugs. And we'll try to understand what enabled these advances and what are some of the outstanding problems and how can we tackle them. So one of the popular narratives is that data and model scale has improved model quality and given these models new capabilities. So in just a couple of years, on the language side, models have increased 1,000-fold, for example, from a model called BERT with something like 340 million parameters to models like PaLM and Gemini with 500 billion parameters. Not only do larger models get higher quality, but they also can give us new capabilities. We can also get new capabilities by not only scaling in parameters, but also by scaling in the context length. So for high resolution imaging or modeling audio, video, DNA, we really want to be able to support long contexts of input data. We can also do well by scaling the number of samples that we generate. So suppose we have a relatively weak model and we sample something like 100 to 1,000 times, we can see that models such as a 70 billion parameter llama model, which many of you might have heard of from Meta, can perform as well as something like GPT-4o on challenging tasks like code generation and math. So the main question that's interesting for us is, can we do something similar for medicine? Recently, there's been an explosion of DNA data. The cost to sequence DNA is dropping dramatically from hundreds of thousands to hundreds of dollars over past couple of decades, and it's becoming an abundant source of data in biology. However, there's a long way to go to really be able to fully understand and compose and create new structures with it. And overall, while the progress is astounding, one of the main fundamental challenges that limits our potential is that our core ML architecture and modeling primitives are quite inefficient still. So a key bottleneck in machine learning is compute. The transformer architecture is the main architecture that powers industry models, and it requires the compute and the memory resources to grow quadratically with the input size, precluding dreams of modeling, say, millions of lines of code or 3.2 billion nucleotide pairs in a human genome sequence. So yeah, again, during training, attention scales quadratically in the input length. And during inference, the memory requirements scale linearly, and thus presents a massive resource requirement on the GPU. The MLP, a different part of the transformer model, scales quadratically in the model dimension. And the scaling gets even worse if we want to support complex tasks that are outside of the transformer's complexity class. So in medicine, many of the types of things we would like to do, again, require long context reasoning. So we're starting to see these models applied to modeling human genomes, performing clinical report and medical video Q&A or medical dialogue, and even modeling time series, for instance, patient vital signs, respiratory rates, heart rates, oxygen saturation, and audio. And all of these really require much longer sequences than many of our popular models are able to support. And there's been tons of great work on genomic foundation models, but historically the context lengths that have been used are fairly short, capturing just a tiny fraction of the human genome. Also, foundation models take in the raw text and often tokenize it, controlling the resolution at which we're going to model. So in text, we're not operating typically with our popular models at a character level, but instead we break up words into something like 50,000 different subtokens that we'll then model over. We really want to be able to have fine-grained sensitivity for modalities like DNA, where small changes to the nucleotide sequences can be really important. And so, the sensitivity at which we're operating is really important. So context matters because it can really help us model much more complicated sequences. Our team, so my lab mates at Hazy Research, also advised by Chris Ray, have, with some of the innovations that I'll talk about throughout the rest of this talk, we've had some really exciting breakthroughs on the medical side as well. So these folks put together one of the largest DNA pre-training data sets, 300 billion tokens of raw DNA, with no real annotations besides knowing the species type. And then we can sample from this data set some random sequences as step one, and simply train the model to predict the next nucleotide in the sequence, and do this billions of times. So for many of us who've used GPT in the way that these models generate, one token at a time, we're going to try to apply the same training objective for DNA and see if the model can learn the underlying distribution and statistics of the data, and interactions between DNA and RNA and proteins. And then the idea is to take the learned model and apply it to the prediction, apply it to predict and generate samples to probe what's learned. Okay. So we with Evo, we've been able to train models with drastically longer context than prior biological foundation models, and start to see some really interesting and exciting quality on standard benchmarks. Just not only on standard benchmarks, though, but we've also been trying to see if we can prompt these models to generate early AI-generated CRISPR systems and genome sequences. Again, via prompting. But despite these really amazing early results and inklings of magic here, we're still in the very early innings of AI for biological modeling, and there are tons of limitations and mistakes that these models are making. We really need a bunch more systematic study, including analysis into how these models work and validation of these models' generations. So for the rest of this talk, I'm going to dive into some more technical depth of how we've done this systematic study I'm talking about on the language side. And my main hope from showing this to you all is that with your medical expertise, we can start to bring the same types of technical tools and methods that I'll discuss today to the development of biological foundation models as well, towards getting these models ready for action. Again, many of the slides I've shown at the beginning of this talk are by, a broad set of lab mates and collaborators. So Eric and Michael Polly really spearheaded the hyena DNA and EVO work. So I really encourage you to, reach out to them if you're interested. Michael Warnow here is also working on foundation models for modeling clinical reports. And so, this is work by a broad team. Specifically today, again, I'll tell you a story about one specific research question. Can we remove attention for language modeling? And what are the consequences when we go from this expensive attention primitive that I talked about that scales quadratically in sequence, has length, has very expensive memory requirements. And what happens when we replace that and go to a more efficient alternative that can better help us model longer sequences? And there's been in the wake of rapid model scaling, actually a Cambrian explosion of these new efficient language modeling alternatives to attention. There are many, many new models popping up these days, always accompanied by some sort of mandatory animal mascot generated by Dolly, if you all are on Twitter, that accompany their announcements. And excitingly, that a lot of the recent work claims that these new efficient alternatives to attention outperform transformers everywhere. So maybe replacing attention, getting long sequence modeling is a solved problem. But let's dive in. Again, this is the picture from prior work. We can take away attention, which is the workhorse of these transformer models, get these new efficient LMs, and still have great quality. But despite the claims of these prior works, a lot of the organizations training the largest and strongest language models are still sticking with the transformer architecture right now. And it makes sense that folks are reluctant to leap away from transformers because the classical saying in ML is, there's no free lunch. When we get something great, like way faster training and inference, long context modeling abilities, we might be giving something up. And prior works don't really highlight what that is. So it's really reasonable that people are waiting to dig in before until they know the price of the meal. So in this talk, we want to find out more about what we're trading off when we move from these tried and tested transformer models to these more exotic, faster, newer architectures that can help us achieve new capabilities. And what are the trade-offs that they're making? For the rest of the talk, I'll explain why these efficient alternative architectures to the transformer are so fast. I'll define the prevailing technical approaches there. I'll next dive into how we went about addressing this complex question of how to evaluate the quality of efficient architectures relative to transformers on language. I'll then show that despite the types of ways in which prior work has presented this problem, there are still very real gaps between these efficient architectures and transformers, and some of these might have very real consequences for biology as well, although I'll ground my discussion in language. My goal is, again, to convey to you tools, including theory, synthetic tests, and evaluation strategies that can help you reason about how new efficient architectures work as they pop up in the landscape. And finally, I'll show you some simple ideas of how we can use our understanding of these gaps to build better efficient architectures. All right, so let's jump into the prevailing approaches. Taking a step back, I mentioned some of the limitations with attention, but let me just really hammer in why it's so slow. The main issue is that GPU memory consumption for attention grows linearly with the length of the text or input sequence. This limits again our context length and the batch size. So with attention, our working memory means that as we have some input text, we need to store what's called a key and a value representation for every single token in our text. So during generation, when you're using ChatGPT or something, we're maintaining this KV cache in GPU memory under the hood that is growing with the sequence length. And each of these tokens in the text is represented and stored with a D-dimensional key and value embedding. The alternatives are much faster because instead they consume a constant amount of GPU memory that's independent of the sequence length. There are two primary classes of efficient architectures, largely falling into what's called a linear attention and a state space modeling architecture. To make sure we're on the same page later in the talk, let's quickly dive into each of these classes. So with standard attention, again, this workhorse of modern ML, what we do is take some input representation of our text. So suppose our sequence length is length n, and we're going to represent each of our tokens in some d-dimensional space, giving us this u in an n by d space. We are going to multiply it by some learned weights, WQ, WK, WV, to get what's called queries, keys, and values. And then in attention, we compute our outputs Y given some exponential of queries and keys multiplied, and then our values. So when you look at these queries and keys multiplied, what you should see is that we're effectively multiplying one copy of our input with another copy of our input projected in different ways. This really allows every single one of our tokens in this n long input to interact. And this QK transpose result, the resulting matrix is n squared. It's n by n. In linear attention, we generally replace this exponential function around the QK transpose in this quadratic matrix with some alternate function phi. So we can multiply phi by q and phi by k transpose first. And the main idea here is that rather than having that QK transpose matrix that scales quadratically in sequence length, we can change the order of the matrix multiplies in linear attention to get scaling that's linear in sequence length, which is much better if we want long context modeling. During inference, in contrast to the KV cache that I mentioned for attention, where we need to store all the keys and values so that we can multiply with our queries first, by being able to reorder the matrix multiplies in linear attention and multiply keys and values first, we can actually just get away by storing a constant size state during inference called the KV state, which, as I'm showing here, h sub t has dimension 1 by roughly d squared. So something that's independent of sequencing. So that's linear attention. The TLDR is we get linear scaling in sequence length and constant scaling during inference as opposed to the KV cache. Another popular class of these efficient architectures is built around a primitive called state space modeling. And this is a concept that's something discussed in Electrical Engineering 101 class. So hopefully it's not too intimidating, and we'll try to just simplify it a little bit. The SSM model is based off of two main equations. So again, like the very similar to the structure that we saw for linear attention, we're going to have this recurrent style update rule where we're maintaining this h sub t recurrent state that has dimension independent of n, independent of sequence length, and only dependent on model dimension. And then using that h sub t in the second equation, we can generate our output y sub t for a given time step in our sequence. And here t represents the current time step that we're at in our sequence. For SSMs, recall that for the attention models, we typically generate our queries, keys, and values as functions of our input u, and then perform our computation. But for SSMs, these A, B, and C matrices that are really parameterizing our two equations here are going to be learned and fixed weights at model train time and are not typically functions of the actual input. And this presents really real consequences. Just to foreshadow, a little bit later in the talk. So state-space models, like the linear attention models during inference, allow us to just maintain, again, this constant size h sub t. And for language for large scale ML, we want models that are really also parallelizable during inference. We don't want this very sequential update rule of h sub t minus 1, h sub t, and so on as we're trying to train these models at scale. And so what's really nice about SSMs is that when we look at their equation, we can actually rewrite it in terms of another popular ML operation called a convolution by parameterizing the convolution filter using these fixed A, B, and C matrices that I mentioned. And a convolution written in like a matrix form has this very structured way in which the n tokens in an input get to interact with each other. So in contrast to attention, where we had this q, k transpose defined in a very input dependent way, so our queries and keys are functions of the input, resulting in a very flexible way that tokens get to interact in this n by n matrix. Convolutions have this very fixed, structured way in which tokens get to interact. But with structure comes, efficiency. There are n log n ways of being able to compute the structured matrix for the convolution, but again, attention has n squared scaling. It's really important for us to, sort of define these different efficient models because some of these foundation models like EVO and Hyena DNA and models that are able to get long context are powered by these alternative architecture primitives like SSMs. So to summarize where we're at with SSMs, we can also get this sub-quadratic, sub-n squared scaling during training with the convolution. And during inference, we can again have this constant, h sub t recurrent state that we get to maintain, as opposed to the expensive KV cache. All right, so we have some different alternatives to attention, but how do they fare compared to transformers, which really power ChatGPT and Llama and all of the main models? So this is a very complex question because language modeling is a very complex and broad task with many different subskills. So we took a very, very simple step to get started in this question of just training a bunch of these different efficient architecture proposals on the exact same standardized setup. So the exact same training data, same infrastructure, and looked at their perplexity, which is the main metric in language modeling. And perplexity basically summarizes how uncertain is this model about what the next token in the sequence should be. And so lower uncertainty or lower perplexity is better. Clearly, there are some gaps. So attention has a much lower perplexity. And although 1 or 2 or 3 points here seem small, these are exponentiated values. So a point of perplexity really means a lot in terms of quality. So why do these gaps exist? We performed an error analysis of the model's next token predictions in a quite manual way. And again, language modeling is very complex with many, many different subskills. So it's completely non-obvious that we would find anything meaningful in this very manual brute force error analysis. But surprisingly, when we did this, a single glaring failure mode for the efficient LMs emerged. So let's look at an example of it. Here, I'm giving you an example where we're looking at how attention and non-attention efficient models performed on next token prediction for a specific language modeling sequence in the test set. So focus your eyes on Pixar Fest, this bigram or series of, it's a pair of two words. The first time that bigram appears, both attention and the efficient models are incorrect in predicting what that token is. But the second time, in line 4 here, we see the word Pixar, and both again are incorrect in guessing that. But to predict best, attention is correct and the efficient LM is wrong, demarked in blue here. What really needs to happen is that when we present a model, some sort of sequence in context, whether it's our language modeling data or some biological sequence, we need our model to be able to go back and look at patterns that it saw in this context to be able to generate the next token if it's a strong and generalizing model. And this specific skill of being able to, go back and pull forward information like predicting Fest as the completion for Pixar Fest is referred to as associative recall in the language modeling literature. This is a one-off example that I gave you. We really wanted to measure this phenomenon at scale. So we took all of these bigrams, so Common Buzzard, Pixar Fest, and looked at, when is this bigram appearing repeatedly in the text? And also, when is this bigram really rare in the training data? So not something like, say, White House or United States that the model might have just memorized, but something that really forces the model to use the input context and refer to information only presented in the input context to generate the next token. So what you can see here on these charts is that we've put this occurrences in the training data or training data frequency on the x-axis. And when we focus on these bigrams that are appearing really rarely in the training data, so towards the left-hand side of this left-hand plot, the gap between efficient models like Hyena or Hyena DNA and RWKV versus attention grows really large. Again, lower perplexity is better, so attention here is basically doing this recall task extremely well, while these efficient models are massively struggling. We found that this associated recall issue accounted for 80 to 100% of the quality gaps on average between roughly 10 different efficient model proposals that have been popular in the literature as compared to attention. And this is, again, really surprising given the number of different subskills in language modeling, that really a bunch of the issue is just localized to one skill. These gaps and these limitations were really surprising given all the discourse around these models, not only from academia with models like Mamba and Hyena, but also with industry. And so Google also has this paper here, Infinity Attention, where again, it's claiming that you can do infinitely long inputs and have perfect recall scores. So to study recall and associated recall in a principled manner so that we can understand why these issues occur and try to build better models, many works have actually abstracted the real world recall problem into synthetic tests. So one example of a synthetic test is here, where the model sees pairs of key value tokens, like C maps to 8, A maps to 3, D maps to 1. And then the model needs to use this information, these random mappings seen in context, to generate answers to queries on the right-hand side. So the model, for example, if we give the query A, needs to predict out 3. What was more surprising than 2 slides ago, where I showed that all these works are claiming that we have these perfect efficient models, was that a lot of these works actually specifically showed that these efficient models could solve associative recall perfectly. This heavily contradicts our downstream analysis on the real language modeling data compared to these synthetic tests that these folks are focused on. we really needed a precise formalization of the recall problem to make progress in explaining these discrepancies and to explain what's going on. So this is where some of my work comes in. And beyond the perplexity metric and overall, those next word prediction language modeling, examples that I gave, we can also look at real world evaluation benchmarks. So a bunch of these models and papers in the language modeling side do try to show how the models perform on real, downstream reasoning benchmarks. But a lot of these benchmarks are only very short context and more classification style, not really testing the associative recall behavior. When we focus on, in my work, really stress testing these models on long document QA, information extraction, tasks that are also very important for analyzing clinical reports and so on, we can see that the gaps are very, very stark. So H3, a model closely related to hyena, and Mamba, which has been a very popular model, the gaps to attention on these types of recall tasks are just, very stark. So let's try to build some intuition on why these gaps are occurring so that we can develop some insights for, better alternative architectures. Let's start with just an intuitive explanation for the first part. All right, so to solve the associative recall task, and again, here I'll focus on the synthetic form of it, a model needs to be able to look back at tokens seen in the sequence to find matches, so which token pairs in a sequence match, and then needs to be able to bring forward the completion for the mapping to generate the next token. So if our query is A, and then we need to fill in the question mark, the model needs to go back and map, one token or two tokens prior to the prior A, see that the mapping is 3, and then pull forward and shift forward this value 3 to generate this token output. Attention makes this extremely easy by computing the full and squared matrix of all token-to-token interactions in the sequence of length n. So again, when we multiply these queries shown, I'm not sure if my cursor works, but yeah, queries and keys, we can get exactly the position of the matching tokens and then be able to pull forward the maps, the values that they map to. But as I kind of foreshadowed earlier, these SSM models and these convolutional models, efficient architectures, are modeling in much more restricted ways. They don't permit easily identifying the locations of these matching tokens in the sequence. So with this very structured matrix from these convolution models, a token can only actually look back m positions in the sequence if every single token looks back m positions in the sequence. Again, due to this restrictive structure. This is not ideal because, as we know in real language or any sequence, there are many different types of tokens and positions at which there could be matches and a need to perform associative recall. So if we can only really go back and compare to one token distance prior, this is very limiting for quality. Luckily, our convolutional models and our SSM models are actually applying a unique convolution at every dimension of the d-dimensional input. So we can support multiple different comparison distances. But if we want to support all the n-squared token interactions that might occur in real sequence modeling, we would need to perform all the different distance shifts, which means that we need the number of convolutions that we use or the total dimension of our model to grow with the sequence length to be able to store a full copy of the input in a single embedding. This is not ideal because our sequences can, again, be very large and very long. We would definitely not want this, this scaling where dimensionality is growing with sequence length. To reiterate, attention does not have any such scaling. It's able to solve recall perfectly with constant dimension, independent of sequence length. All right, so To start to make progress in understanding the recall problem, we provided an improved formalization of the problem. So what is wrong with a lot of these synthetics that people are using, across industry and tons of academic work, is that we're really only thinking about vocabulary sizes of up to 50 tokens and very long sequences. So the types of ways in which prior work was stress testing these models meant that in a long sequence, if you only have, let's say, 50 different types of tokens, you're going to get these key value mappings repeating many, many times in the long sequence. So we might see A maps to 3, D maps to 1, 10 times, 50 times, 100 times. And so a few convolution shifts are sufficient to kind of cover all of the unique different key value mappings that we might see. We put together an improved synthetic formulation where much more akin to real language modeling, where the number of unique subsequences or key value mappings in the synthetic language are much larger than the total dimension. We made it so that the vocabulary sizes were much larger. You're only seeing each key value pairing, say, one or very few times. And so the model really does need to be able to support a broad range of shift distances to succeed at the synthetic. And indeed, when we plot how attention versus these efficient models perform on this new improved synthetic as a function of their model dimension, we can see that attention gets perfect accuracy on the synthetic at across the board of model dimension independent of sequence length. But indeed, these efficient models like Hyena and H3 are requiring the model dimension to grow with sequence length in order to get good quality. Again, this correspondence between the model dimension and sequence length for these efficient models is very, very undesirable and inefficient. But in the literature, there are actually many, many different variants of these efficient subquadratic models. And so a natural question that somebody might ask is, do one of these other candidates out there fix it? So in my work, we then turn to a more theoretical way to reason about this massive landscape more systematically, in contrast to the sort of intuitive demonstration of how a model might solve associative recall that we just discussed a few slides ago. The main ideas here, just to flash a couple of the technical nuggets that support this work, are that we can actually write each of these efficient models as computing some sort of polynomial. The primitives that underlie these architectures are things like element-wise multiplication or a Hadamard product, or a convolution. And each of these is actually computing some sort of polynomial operation. And we can unify the broad, messy landscape of architectures using this lens of viewing these models as arithmetic circuits. And in math and theoretical computer science, arithmetic circuits are a very natural, powerful computational model for studying the complexity of polynomials. So when we can write our models as polynomials and we can transfer them to these arithmetic circuits, we can get these nice equivalences between these broad classes of architectures within some theoretical bounds. So I won't get into the details for the purposes of this talk, but the TLDR is that we can take this messy landscape of efficient architectures that are being proposed and distill them into this very concise, what we call a base conv layer that essentially has the same exact scaling on these associated recall problems as any of these other diverse architectures shown here. And using this, theoretical work, we can show a lower bound that any of these convolutional SSM models will require the dimension to scale with the sequence length in order to solve associative recall. So we really do need something else as opposed to these SSM primitives that we've been discussing, and which also, again, powered models like EVO and Hyena DNA. Again, the summary is that attention gives us really nice, flexible, input-dependent sequence mixing. The convolutions are much more restricted. So to build a better efficient architecture, we would like something that's actually, still going to be input dependent and flexible like attention, but avoids the quadratic cost of this expensive QK transpose matrix that I mentioned at the beginning of this talk that limits attention from doing long sequence modeling. So let's, towards building up our intuition for the next part of the talk on how we can close the gaps, let's turn back to the synthetic, associative recall task that we, built up. So attention, again, gives perfect associative recall accuracy, but has a huge state requirement or huge memory consumption as shown on the x-axis. A very, very simple baseline in ML is that is also input dependent, but doesn't require the quadratic scaling is something called a sliding window attention. So the main idea here is that rather than having every single token interact with every single other token in the sequence, resulting in n-squared scaling, let's have every token only interact with a prior fixed small sliding window of tokens, say like 64 or 128 tokens. As we vary the number of tokens that get to interact with each other on the x-axis here, this changes the memory requirement of our attention, but also drastically impacts our recall quality, presenting this very stark trade-off between quality and efficiency that's rather undesirable. As predicted by our theory and shown in the bulk of this middle part of the talk, these gated convolution or SSM models fall below the frontier even of sliding window attention. And again, very undesirable. Recent models that you all might have heard of on the ML side, like Mamba, were able to expand the Pareto frontier beyond sliding window attention for the first time. And to do this, they add more selectivity to the standard default SSM modeling structure to make it less restrictive and more input dependent like attention. But fundamentally, from theoretical computer science, we have really fixed lower bounds that any model that uses less memory than the full sequence, less memory than like scaling in n or the sequence length, is going to have to sacrifice in recall quality. And this is very intuitive. If we have less memory than the entire sequence length, then we're not going to be able to remember all of the information in our very long contexts. So there is this trade-off that's very fundamental here. And the whole game is how we can get models that better expand this Pareto frontier. To reiterate, the differences in the size of the working memory. So our efficient alternatives use a constant amount of memory independent of sequence length, while attention, again, uses memory that grows linearly with sequence length. I'll briefly touch on a couple of techniques from my work on how we expanded the Pareto frontier, far beyond architectures like Mamba. And these are, some emerging and growing architectures in the space. So alongside trying to make these efficient models have better quality, work is taking increasingly steeper and steeper departures from standard attention into much more exotic primitives that might not actually compute all that well efficiently on modern GPUs, and might be difficult for different people to adopt because they're rather complicated to understand. So my work asked if a very simple thing could work. We're just going to take linear attention, something we've discussed a lot in this talk. It's input dependent like attention. It's still sub-quadratic in training and independent of sequence length in its memory consumption during inference, and gives this nice global approximation of standard attention. I mentioned that for linear attention, we are going to replace the exponential function in vanilla attention with some feature map function phi. And so what all I'm going to do is say, let's take a Taylor approximation of the exponential function and let our feature map function phi be this exponential function. This might go over heads. We don't have a ton of time to discuss this, but the main idea is just we're going to replace the exponential around QK transpose with some feature map function phi that's very, very natural and direct following from the exponential function. And all we're going to do, because we're combining this coarse-grained linear attention that's only a global approximation of attention, is to add a dense sliding window attention that is actually using the softmax and using the original attention structure, but only in a dense local sliding window, which I mentioned can be much less memory intensive. And when we combine this very, very strong local mixer that allows tokens that are nearby in a sequence to densely and tightly interact with each other with the global linear tension that supports long-range dependencies, we get our architecture here, which we called BASED. And BASED was able to expand the Pareto frontier of this memory and recall trade-off space beyond any of the prior architectures. And using, so I'll skip through some of these slides, using our innovations on the efficient architecture and CUDA development side, as well as these architectural developments, we're able to close much of the gap between attention and prior models like Mamba and Hyena and so on, to get much better recall qualities and overall language modeling qualities retained at the same time. We've been really excited to see these ideas in our new efficient architectures getting used. And again, the call to action here is that for these AI foundation models, the DNA foundation models, and so on, we're really just very much in the early innings, and we need such systematic analysis and synthetics and theory and evaluation work to get these models ready for prime time. Right. Yeah, so those are the key takeaways, and with that, I'll stop here and take any questions, and thank you for listening.
Thanks so much for that, Simran. It was really awesome. I'll open the floor to questions. If anyone does have, feel free to unmute yourself and ask a question. I guess while people are doing that, I think one thing that would probably be very useful, I think, for the audience is what would you recommend to them is the way to get started, especially with these sorts of like architectures for them to like really just get their hands dirty, etc.
Yeah, that's a great question. So to evaluate a lot of these architectures on really massive scales is difficult on a academic budget without many resources. And so I really believe in trying out these synthetics. That's actually the way that I got started with a lot of these resources, just taking some of these models that have been proposed and running them on these toy tasks, these simple synthetics that you can actually get results in a matter of minutes and use on very, very cheap GPUs. And our work we've put up a bunch of repositories that I'm happy to share with anyone if they reach out or email or if they look up on GitHub, where we've pre-implemented a bunch of these efficient architectures and these synthetic test beds. So all it requires is sort of, cloning and plug and play and very easy to get started.
Awesome. That sounds great. And I think related to that, I think Prinsai has a question in the chat if you'd like to unmute both. I think on links and your second question.
Try to find the chat.
Yes, I could.
I'm not seeing the chat on.
I think Ras, I think you are unmuted,
I
right?
have a question.
Yes.
I don't know if you mentioned my names. All right.
Yes, I did.
Okay. It's so thanks for great talk. It's really nice. Although I'm not so familiar with what you do, that's why I think this is quite a simple question. I'm just curious, why all these models? You mentioned lots of. animals' names? I mean, why is the inspiration? There is some connection, or why? And second, yeah, if you have some applications of these models in medicine, especially in rare diseases, if you know, or how can be applied or some things.
Yeah, so that's a great question. So we just go back to, the beginning where we kind of motivated a bunch of these. So the main applications in medicine right now, we're seeing in, medical Gemini with medical video question answering, DNA foundation models. The desire to have longer sequence lengths is what's really motivating the use of these models in medicine. So for yeah, so the main reason to look at these architectures is just that attention, which is the de facto architecture, scales very, very poorly. And this zoo, all these different animals are pieces along the research trajectory on the ML side to get models that can scale much, much better if we want to support inputs to models that are very, very long, like long videos where there are multiple time frames, or long sequences of patient vital signs, or long like human genomes. To answer your question on like where these are being used, I'd say that like I'm more of a ML person, less of a biological person. So maybe many of you all know better, but on our end, we think like a lot of this is very, very much in the early days. And with models like Evo and Hyena DNA, we are starting to see these models saturate existing benchmarks, but I think there are needs that we're facing for much stronger and realistic benchmarks. And one of the main challenges is actually taking the generations from these models and understanding, are they good in practice? And a lot of that requires going to the wet lab and working very closely with biological experts. And so there's kind of this interplay between developing these new architectures and also understanding how well they can actually support these intended tasks.
Thanks a lot. Is it possible to share the slides and the links and a way to contact you? I know you mentioned some articles, papers there. So yeah, I would like to follow up. Thanks a lot once again.
Yeah, for sure.
And then I believe Lisa has a question. You'd like to unmute.
Thanks a lot. It's a wonderful presentation. I really enjoyed this. And is there any way, I mean, do you have any tutorials or other means of conducting the knowledge sharing base from your center?
Yeah, we have many, many open source code repositories that are very, very easy to use. So I'm happy to send those out to folks after this. And these models are, what's directly used in some of these larger DNA foundation models that I mentioned as well. And our lab is also working on clinical foundation models based off of these. So foundation models from clinical reports based off these.
I'll be pleased to know the means which I could contact and learn more on it.
Yeah, I'm not sure the best way to send out my email address, or I mean, it's SIM, my last name, at stanford.edu. But yeah, folks are welcome to reach out. Yeah.
It'll be appreciated if you could text it here.
Yeah, let me try to do that. Struggling to do screen sharing and my yeah, sometimes Zoom doesn't show the bar. Okay.
Yes, and then I believe Lisa has a question.
Oh yeah, thank you for the Simran, for the good talk. It's really kind of not only enhance my point of view, but how do we can take measurement models to be more engaged with the clinician's perspective. But also, I have a questions. How do you think we as a scientist or researcher or perhaps PhD students to make or create the best approach to the clinicians in how to making foundation models? I mean, because as you may know, until this time, there are so many models using machine learning or deep learning or what is it usually we call or perhaps MML, but it still not cover the request or or the requirement from the medical context. So what do you think the best first approach for us to do that?
Yeah, that's a great question. So a lot of the medical tasks right now are very application specific. So what's really important, I think if more of the medical experts could provide and like work along with ML folks is what are like the skills that a model needs to have to be able to succeed at these tasks. So if we give a model, 50 different benchmarks, that might seem amazing. Like we've curated all these different evaluation benchmarks, but if they're all just simple classification, like, what species is this DNA sequence from? Then we're not actually really testing a broad range of reasoning capabilities from these models. So we need a better bridge between applications and the specific modeling capabilities that might be required in a way to like break down that taxonomy. And in a lot of our discussions right now in this work, we're just totally missing that and totally missing, there's kind of a lack of communication between biological experts and ML folks in curating these evaluation benchmarks. So intuitively, maybe recall is something that's very important for, even DNA long sequence modeling. But if we build all these benchmarks that are only classification style, then maybe we're not actually stress testing the importance of some of these skills. And
Yeah,
there are many
thank you.
more skills beyond recall. Yeah.
Yeah, because I think there's still a huge gap between us and the clinicians. Yeah, thank you. I will try that during my research. Thank you.
Then we've got another question from Yaman, if you don't unmute yourself.
Hi, thank you very much for the great talk. My question is more technical, more on the comparison between attention-based transformer and the convolution. My background is in imaging, but also recently I have been looking into NLP and the sequence-based modeling, and so I'm wondering, like when you have convolution, can these convolution-based models be used for generation, for example, like generating the next token? When you do that, how do you apply the convolution? And if I understand correctly, are you applying any masking in the convolution as well, or like for images we have the convolution kernel, and normally for images the kernel is small compared to the image size or compared to the sequence length. So the convolution, the kernel size in those convolution models, is it the same length as the whole sequence, or it's smaller than the whole sequence?
Yeah, these are excellent questions. So let me just go to the SSM slide. So we mentioned slide takes a long time to iterate through. I just want to show the SSM equation as I talk through this. Okay, so what I mentioned is that there's this equivalence between this recurrent. So you asked two questions. So one, how do we generate the next token? And then two, how do we handle causal or next token prediction from left to right? So for the first question, the main idea is that there's actually this equivalence between the convolution and this recurrent view that we can see here when we look at the equational forms. And this is really nice because during training, When we want something parallel, like a convolution, we can go with that view. And during inference, when we want to generate one token at a time, we can compute the outputs using the recurrent view. So mathematically, these two ideas are equivalent, and one ends up being better than the other, depending on whether we're in inference or in training mode. To answer your next question, so exactly as you said, in images, traditionally, the filters are very short, something like 3, 5, 7. For these SSM models, these convolutional models in language, we really are using a filter that is as long as the entire sequence length. And that's really emphasized here, where you can see CB, CAB, all the way up to C a to the n minus 1b. So that this filter is actually yes of length n. And if you were to naively take an input of length n and have a convolution of length n and try to apply that, it would be non-causal, following your intuition. For other folks, our standard language modeling objective is that we hide the future tokens of the model and ask the model to generate and predict what comes in the next spot. So we need to be able to hide what comes later from tokens that are earlier in the sequence. So to get around this, you can just simply pad your convolution filter, or double the length, so that when you're applying it, prior tokens only see the padding and don't actually see the future token. So it's a pretty standard technique for causal convolutional application. I'm happy to send you code, but the main idea is that we can just pad the filter.
Thank you very much. Thank you.
And then we have a question from Anna. You'd like to unmute?
Yeah, sure. Can you hear me well?
Yeah.
Yeah, okay, great. So I've got a question about combining different modalities. Because I'm coming from biology, I'm interested in like single cell biology, and now there are a bunch of foundation models also used in this area. And also recently there have been some publications about combining imaging and genomic data. So I was wondering, do you know any like state-of-the-art applications or algorithms that we can use for combining this quite different modalities, and how shall we tackle this like difference in modalities?
Yeah, that's a great question. One of the really exciting works that came out lately that you could take a look at is like this transfusion work from Meta, Facebook. And like the main idea is to take like patches of images and patches of tokens in the sequence and to model these all in a unified way. So I would check out that work. I think it's like one of the most promising ones that I've seen lately. But yeah, I don't have a ton of context on multimodal models in my work.
Okay, thank you so much.
And if there's, I guess, no other questions, allow me to thank Simran again for a really amazing talk and for making the time to join us super early out on the West Coast.
Of course. Yes. Thank you all. Yeah, thanks for having me. Did I stop sharing screen? Sorry, I was just trying to
I think it's still sharing.
still sharing. The Zoom bar is elusive.
Yeah.
There, I see it. All right.
Awesome.
Yeah, thank you so much for having
Thank-
me and for all the
awesome.
great questions.
Awesome. Thank you so much, Simran. And I guess with that, everyone, it brings to a close this year's 2024 CCAIM Summer School. Like, before I hand over to Andreas, just from my side, to thank everyone for joining us, for all the the amazing engagement. Like, the summer school isn't anything without the participants, and thanks everyone for making it a really great summer school. We hope everyone's enjoyed it. And before fully handing back to Andreas, I think I just want to actually acknowledge both Andreas and Miha's efforts on the summer school. Like, I think they put in a huge amount of effort behind the scenes, both before the summer school, making sure everything was in place and running it seamlessly this week. So I think all of the success is really down to their efforts. So thanks both, Andres and Mia. And I guess back to
Thank you,
Andres.
everybody. Thank you, Nabil, and thank you to you too, because you had a lot to do with organizing our summer school. I wanted to thank everybody for coming, and I hope you enjoyed it. Thank you for your presentations as well. This is something that each year it's very interesting to see. And thank you again, and all the best. See you next year. Back to Andreas and Nabil. Thank you again for everything, for all your efforts. Bye, everybody.
Thank you. Bye, everyone. It was great summer school. We will make sure to send you the certificates in a couple of weeks. So stay tuned for that and the last recording this evening. Thank you very much.
Thanks a lot. Bye.
Thanks all.
Thank you very much. I enjoyed it a lot.
Thank you so much. Great event.
Thank you. Means a lot.